Good morning, Dahika.
Yes, sir. Good morning.
Good morning. Thank you. And now, can you see the video? I can see the screen from my laptop. Could you see my video also? Is it the window?
Yes, sir. Your video is on. I can see you.
Thank you. Okay,
and your
that's great.
PPT
Thank
also.
you.
Yes, sir.
Okay, excellent. That's great.
Yes, Rachna, you can start now.
Yes, sir.
Yes, Rachna, madam. Please proceed.
Good to all, and a very good afternoon to all the excellencies participants present here. So it gives me an immense pleasure to welcome you all for today's third international webinar series 2022 on industrial insights of microbial technologies for sustainable protein manufacturing, that is mycoprotein production, organized by Sanjeevani Arts, Commerce and Science College, Kopargaon, in association with Microbiology Society India. So today for this session, we have with us eminent speaker for today's session, Dr. Patenatur Rahman, Senior Lecturer in Bioscience, School of Pharmacy and Biomolecular Sciences, Liverpool, John Moores University, Liverpool, United Kingdom. Also, we have today with us respected Dr. A.M. Deshmukh sir, President of Microbiology Society India, respected Dr. S.B. Dahikar sir, Principal of Sanjeevani Arts, Commerce and Science College, Kopargaon, Dr. S.A. Buttada ma'am, Head of Department, Microbiology, all staff members and all the participants present here. I once again welcome you all for this international webinar. So first of all, I request our respected Principal Dr. S.B. Dahikar sir to give a brief introduction of today's webinar. Thank you. Over to you, sir.
Yeah. Very good morning to Dr. Rahman sir, and good afternoon to all the participants in today's session. I'm very happy today. This is the third webinar series, international webinar series, organized by Sanjeevani Art, Commerce and Science College, Kopargaon, Maharashtra, India, and in collaboration with Microbiology Society of India. I'm really thanks to Dr. A.M. Deshmukh sir, the President of Microbiology Society of India, in his guiding to us for the all the budding microbiologists, biologists, all the life science students, teachers, and researchers for the India. And this the main objectives of this platform, that is the international webinar, is just to interact with the resource person, interact with the scientist. And we are really good and lucky. We have today's Dr. P. Rahman sir, is very expertise in the microbial technologies for the sustainable proteins manufacturing. And I'm happy to share you that, sir, our Microbiology Society of India is working last from 1994. And we have organized a lot of conferences, webinars, seminars, and different types of social and community-based activities by which we can aware the community what is the role of microbiology in the daily life and how can we aware to other persons, other people also. So basic is the what is the importance of microbiology and how it will be help for our day-to-day living routine. So that is the objectives, and under the guidance of Deshmukh sir and Dr. Patankar sir, we are growing day by day. And this is a very important session now we organized today. I will not take that much time. I guess thanks to Dr. Rahman sir and Dr. Deshmukh sir for being present today's sessions, and sir has accepted our invitation in a very short period of time. So I really thanks to Rahman sir, and I am happy to inform you that sir ki 27 more than 30 participants are joined, and many more are willing to join. And near about 100 students are now in a seminar hall. They are also listening to you, and that means nearby 200 participants are now in our session. So I'm really thanks to sir, and I will over to you, Nagarkar ma'am, for next session.
Yes, sir. Thank you, sir. So now I request Dr. A.M. Deshmukh sir to address this session. Thank you. Over to you, sir.
Yes, 1 minute. Yeah, okay. Today, really, I'm very happy. The reason is that every Sunday or once in a week, we are conducting various lecture series. But among all lecture series, I am very happy with this today's lecture series. The reason is that we are interacting with the eminent scientists which are out of India. They are sharing their knowledge. They are interacting with our student. And as already I have told, Dr. Samadhan Daikar has taken the responsibility, and this is a third lecture in the series. As a president of Microbiologist Society, it is my duty to give all the best to this series. And I am sure students in microbiology from India will enjoy this lecture. Thank you, Rahman sir, again. Thank you. Thank you.
Thank you, Professor.
Thank you, sir. Definitely, we are having a very good response from all over India. So now, without further ado, we will turn the time over the eminent speaker who is expertise in mycoprotein case study. So I request Aditi ma'am to introduce our today's resource person. Thank you. Over to you, Aditi ma'am.
So very good morning to Dr. Rahman and very good afternoon to one and all present here. I am Aditi Deshmukh, Assistant Professor at the Department of Microbiology in Sanjeevani Arts Commerce Science College. It gives me great pleasure to introduce our today's eminent speaker, Dr. Pattanathu Raman sir. He has 25 years of industrial and academic experience in research and development, innovation, management, and commercialization. He is working as senior lecturer at Liverpool John Moores University. Sir is author of 75 international journal publications with more than 7,000 citations and editor of 3 books on the topic of biosurfactant. Dr. Raman completed his bachelor's, master's and doctorate degree at Bharatiya University, Coimbatore, and biologics and biochemistry, Vansar at University of Manchester. Started his career postgraduate in University of Northern Ireland, research scientist at Brookhaven National Laboratory, New York, USA, and head of biotechnology program at Teesside University and University of Portsmouth. We are really honorable to have you, sir. Now, I would like to request sir to proceed the further session. Thank you.
Thank you. Thanks for the nice introduction. And thanks, Professor Deshmukh, and also the organizers from Sanjeevi College. Today I'm going to share some of the experience from my research and also the industrial opportunities around the research. I think it will be useful for researchers or microbiologists, those who are listening today, to those, if they have access to research labs, they might be able to do some work related to this area and to see how it works. And it's an exciting topic. So after got the invitation, so I thought of selecting the topic which is suitable for India. Mainly this area is about vegan protein. Vegan protein, actually India is one of the largest population around the world, mainly vegetarian or vegan food diet. So clearly there is an opportunity from microbiology or industrial biotechnology to provide it's another food supplement or a food source from microbiology. So I think it's in this work I'm going to share a case study that will help, that will give you some idea if you're planning for a research from your master's program, so that you'll be able to see how you can plan or you can identify your area of the research in this sector. So just to briefly introduce, on the beginning they introduced about my research background. So currently I'm placed at Liverpool. So those who are not familiar with the map of UK, so it's Liverpool is in England. It's northwest England. It's a coastal city and it's a beautiful city. So we had a number of like other major cities, various museums, sightseeing places, and University of Liverpool and John Moores University. It's all in city campus. We are based in city campus. So that means our departments, schools will be around the city. So we need to move to different buildings. It's an exciting place. It's one of the best part of UK, I would say, and also Liverpool Football Club is one of the famous football club in the Premier Leagues. So today's topic, so microbial technologies. I'm going to cover some basic information about what are the various techniques we can use and how to design or how to design our approach in terms of sustainability. For example, any of the bioprocess, a lot of waste will be generated, how to utilize that waste to prevent or to make various other products. So I'll highlight some information and also mainly the mycoprotein case study. So the mycoprotein, so I think as a microbiologist, we all know it's related to fungus. So the mycoprotein means it's a fungal protein. So most of you may remember when you go to the supermarket, it's available. It's I think a mushroom. It's quite commonly available. It's also protein source in supermarkets. Whereas if we separate mycoprotein from fungal hypha, or if it's a fungus, which so the fungus, which has a hyphae, that could be used to produce these protein. So how to achieve that? So it's quite a lot of challenges associated with that. Today, I'm going to share some information how we can do that. So first of all, this case study, it is so currently it's moved to large industrial scale production, but it started from it's a local soil. So they have isolated this Fusarium venenatum. It's the fungus from the local soil, and then currently it's sold as a product called Quorn in most of the supermarkets here. It is a fantastic meat substitute, and it has the protein composition and also nutritive values. Really, it's a very good combination of nutritive values associated with that. So that helped to the company to market this product. And the morphology, the fungal morphology lends itself, will give the texture of mouth feels like chicken. So it's like a chicken fiber. So it's a great replacement or alternate food for example, for chicken. So as I mentioned, it is a fungus. It's from Fusarium venenatum. And it was isolated from the agricultural soil, the field of Marlow. So Marlow is one of the places in the UK. And it was selected for the use in the process based on its morphology. Probably, I think most of the microbiologists, those who studied fungus, so they know the structure of these fungal hypha. So they are about probably like 4 or 5 micron width, and the length would be like between 300 to 700 nanometer. 300 to 700 micrometer. So this, with this structure, it's also hypha, it's like a hairy structure. So that when we think about bioprocess means we need to use a large volume of liquid and provide the nutrients required for the organisms, and then provide the optimum conditions. For example, temperature, pH, agitation, and various nutrient requirement for the organisms. And then the organisms start to multiply. It's in a fermentation process. And then when you get a large volume, so how to collect it, how to separate. So these are all the challenges in bioprocess development. For example, when you work in bacteriology, For example, bacteria, they have certain micron size. So they have shape and size, spherical or spiralum or bacilli. So when we centrifuge, we would be able to get the biomass. Whereas if it's a fungal hyphae with nearly 300 to 400 micrometer length structure, and also it grows like a hairy, lengthy structure. So it needs a specific methodology or techniques to separate. And to scale up, not to produce an industrial scale production, we need to think how to achieve it. And the nutrient profile of these organisms, excellent. It's a good protein content and with the spread of amino acids, and it's a low fat, high fiber. So that's fantastic property, because most of the currently used meat, mainly if it's from to simplify, cattle or sheep or goat or any meat, it contains quite a lot of fat also, and then that composition. So we need to think how to replace that. For example, if the fungus can provide that mouthfeel-like structure of fibrous structure, and it's a low fat, then it also provide, it reduces the health implications associated with any of the meat-based product. So we can replace that with the microbiological product. One of the best microbiology-based product or fungal hyphae-based product is this one. It's a mycoprotein. So this hyphae, when you compare this with soya protein or a spun soya or chicken or mycoprotein, so you can see the structure. It's a scanning electron microscopy image. So this image, the mycoprotein, it has almost like fibrous, and it's something similar to other protein source available in the market. One is meat, one is a chicken, and one soya. So that is very important when we consider if any product from our bioprocess microbiology goes, how the customers or consumer requirement. And if you target to replace certain products, then make sure the not just the nutritive value, and also other features which is needed, then only the customer or consumers, they'll be interested to use it. So we need to consider these kind of approaches when you think about bioprocess development using microbiology. So for the fermentation process, so typically we need to start with the liquid-based medium. So start with, it's a process flow diagram. So media preparation, and then the media it's needed for any of the microorganisms. So we need to provide the right media, and also the sterilization, it's a key steps, which the media needs to be sterile, because any other contaminant available in the media, that will contaminate the final product or the bioprocess. So it needs to be sterilized. And then this fermentation process, it's a continuous fermentation, and also it's a airlift fermentation process. It's another approach which is very good. It doesn't consume quite a lot of energy, so that we'll be able to effectively grow these fungal hyphae for a large-scale fermentation. And also there are a few more steps. For example, a degas broth, then heat shock, and then centrifugation to concentrate the paste, and then blast chill, and then, finally the process. It's end of process. Final product is produced. It's another schematic diagram to explain how it works. So just I'll move the screen here. Yeah. Okay. So the raw material and the utilities, as I mentioned, it's a nutrient is needed, and it's a airlift fermentation process. So and also this process goes like almost for each, the fungal or bacterial process, we need to optimize the fermentation process. Sometimes for a bacterial process, it takes only a day or two. Whereas for fungal fermentation, it takes a few days, between 3 to 5 days, or sometimes we need to go up to 7 or 8 days. So we need to study. These are all, it needs a lot of research. When we think or when we plan a research platform associated with the fungal protein, we need to optimize these processes to get the best yield or high yield at the end of the process. So how long it would take, and what are the temperature, pH, agitation, and other nutrient requirement for the process. Then, so we need to reduce the RNA content. So there is a specific process used in this, and the centrifugation, chiller. So finally, it is dispatched. So various, the final product is converted into various forms, and then packed and then supplied to the market. So during the process, quite a lot of waste or liquid waste is generated. For example, here, if you could see, even in a liquid broth, so the hyphae or a fungus uses fractions of nutrient for its multiplication to grow. Then the remaining residual nutrient, which when we separate the fungus or bacteria, so it will be it stays in the cell-free supernatant or the liquid. So that liquid needs to be processed. So they have also looked at various other options. So it contains about nearly 5% sugar. So that sugar can be used to produce some low-value product. So that also the company was looking to look at how to minimize, how to utilize that, so that the waste management or operate, to process that wastewater before it released into the natural system or the water bodies. So they have considered a number of techniques how to utilize clarifiers to use all the the organisms present in the system to use the remaining residual nutrient so that effectively it can be treated. And also the sludge generated from that waste also can be reused. So it's waste management is a key when we consider large-scale fermentation, because large volume of so it's a large volume of waste produced with the low sugar content. So the economic reasons for the fermentation, which is the reason they are operating in continuous culture process. So there are a number of 3 major techniques, I would say, or major operation we could do. One is batch, second one fed batch, and then third one continuous fermentation. Batch process, that means so the liquid or nutrient provided for the particular organisms at the start, and then leave it to use all the the nutrient by the organisms for its multiplication, and then finally when the process finishes, you can stop it and then separate the biomass and the cell-free supernatant. So that's the terminology for a batch. Whereas in fed batch or semi-continuous or fed batch process, so whenever the nutrient values reduces during the fermentation process, we can top up or we can provide small fractions of nutrient at regular interval to maintain the organism's growth. So it applies to both bacteria, fungal, any bioprocesses. Whereas in this process, it's a continuous. Continuous process means continuously we can supply the nutrient and also extract the influent nutrient continuously supplied, and then you need to remove the culture broth for a process. So in this process, so they continuously remove to separate the fungal hyphae and then process it for a final product. And most industrial fermentations are, as I mentioned, it's batch wise, because when you operate in batch fermentation, if any contamination happens, only in that batch they can remove and then they can start the second batch. When we consider continuous fermentation, it's if any contamination happens, so that might affect the quality of the product. So when it is operated in very large volume, the operation cost is very high, and if any contamination happens, it's really a huge loss. So they have to be very careful, mainly the aseptic conditions, and then all the operation should be in clean room, and then only if the operation is clean, and then if there is no contamination, so then it is possible. So, and also I would say that since I have been working now in UK for a number of years, what I have been seeing here, the bacterial or fungal loading generally in the air or in lab environment extremely low. Because I have seen when I was doing a PhD or most of the research notes, education at bachelor, masters, or MPhil or PhD in India, always contamination is a big problem when we conduct research.
तो फिर गवर्नमेंट मैम को बोलो, उसको बोला मैंने, गवर्नमेंट मैम की परमिशन नहीं तो पीएच पेपर लेके जाओ। अरे वहाँ पे पेपर है, वहाँ पे एक्सपेरिमेंट थोड़ी करके बैठेंगे वहाँ पे।
So if someone have the access to if you could help to mute that, yeah, that would be great. Thank you. So, yeah, as I mentioned, it's in India. Contamination is huge because of it's a tropical weather, and working in the labs. It's always if you're a microbiologist working with a number of bacteria, always you'll get fungal contaminations and so on. Whereas here it's very minimal. In our labs, most of you will be surprised. So sometimes we don't, not mainly for undergraduation level. Now, if we have a large number of students, so mostly it's operated or worked in aseptic condition. But the labs it's extremely good. It's a clean facilities maintained. But not rarely they use laminar flow chamber, even for plating, spread plate, pour plate, or all the basic microbiology work. But we don't get any contamination at all. So clearly that shows that the air quality. It's the loading, microbial loading in air it's quite low. It's a number of reasons. For example, it's cold weather, so that reduces the organisms or the spore formation and so on. But it's a huge challenge in India. So that means if any bioprocess it's planned in India, then we need to consider very carefully to avoid contamination. So contamination will be a big challenge to any of the bacterial or fungal based fermentation processes. So basically here, even though the air quality is fine, so we operate in lab conditions with the various aseptic technique. So basically you need to be very careful, or else it's difficult to maintain the quality of the products. So that's we need to consider in case of any fungal protein. It's a planned for manufacturing in India. They have to consider this. Then operating continuous fermentation. So that is also poses a number of challenges. As I mentioned, it's a continuous flow of sterile media. That is a key. So you need to have the facilities to supply the nutrient continuously, and it in a sterile condition, and aseptic condition within the fermenter. So that means when we operate in a very large volume, for example, most of the microbiologists, probably you might have seen in the lab, working with probably 50 ml of medium in 250 ml flasks. That's quite common in most of the labs in India. We use that for process optimization and so on. And also we can scale up to 1 liter or 2 liter, even sometimes in shake flask you can do. Then 3 liter, 5 or 7 liter fermenters can be used in the process development. So the so inside the fermenter, so aseptic conditions needs to be maintained because you'll be adding various nutrients continuously supplied, and also if any fluctuation in the pH, acid and alkali additions and various additions are introduced to maintain the operation conditions. So everything should be sterile. That's another key challenge. So how to do that? And also how to maintain optimum conditions in the fermenter. So as I mentioned, a fermenter, it's a very large capacity, but it could be just to simplify that. So if you use a shake flask, so a shake flask is a starting point, but in so we can use shake flask culture as an inoculum in fermenter. So make sure that particular process, make sure it's aseptic. But after for the introduction, there are specific methodologies used to introduce the cultures in fermenter. So all of them needs to be sterile to avoid contamination, and what rate of feed media, and then take biomass of the fermenter. So for example, when you operate in continuous culture, we cannot continuously supply the nutrient without taking the culture, because it will flood, or it will, so fermenters, they have the capacity. So it will fill. So continuously you need to take the cultures and the process for biomass. So that also that needs to be considered. What are the flow rate, inflow and outflow rate? Just to explain this, maybe the organizers, please have a look at if any, any of the participants, if the mic is on, could you please help to switch off that, please? Thank you. So mycoprotein fermentation, as I highlighted, it's an airlift fermentation process. So in that case, in typical fermentation, we use various motors to rotate, or to create the agitation needed for the fermenter. Whereas if we continuously supply ammonia and air, so in airlift process, so if you could see, I hope you can see the cursor. So when you supply, obviously it goes to and then coming down. So it creates micro air bubbles created, and also that itself, it creates like a loop system. So that is sufficient for fungus, fungal fermentation. Probably for a bacterial fermentation, if someone is conducting bacterial fermentation, it may not be sufficient. For bacterial fermentation, we need to optimize probably around 100 or 200 RPM. 200 RPM, and we can increase that, depends upon the oxygen requirement for the bacteria. For fungal process, it is sufficient. It's a airlift fermentation, it works, and the nutrient addition needed, and also cooling system in case of to maintain the temperature, and also when the process, if it's a continuous process, continuously you need to extract the product or culture to process it, like a heat treatment and a dry chilling. These are all the various techniques used to get the mycoprotein. But so not in detail, just again with the schematic, how that can be conducted. So mycoprotein, the fungal hypha production. So initially it's a fermentation, then it needs to go through various steps to create. For example, first you need to separate the fungal hypha, and then steamer to sterilize, or they are operating 600. Some of them, the process changes for certain fungal protein. So in this process, now they are using steamer, and then chiller, then finally size reduction. So concentrate, so remove the RNA content and also any of the residual nutrient. So finally to concentrate the product, and then freezing process used to store it, and then send it to, it's industrial scale production. They have various options before it's put in a packages or pockets. then finally, it's cold storage facilities are used to store it. It's produced in very large volume, like thousands of tons per year now. And it's available in almost all the supermarkets here, and also US and other countries. They are using these fungal protein. It's branded as a corn. This corn product are available in UK market. So, yeah. So just to capture this fermentation process, it's sterile media and oxygen-enriched air are continuously fed into the fermenter, and then media is governed by growth kinetics of the organisms. So if anyone planning for any fungal-based protein or fungal hyphae-based protein research, so you need to carefully, first you need to learn about the growth kinetics or the growth rate of the organisms before doing any detailed fermentation process. So that is that's key. What are the requirement, oxygen requirement or nutrient requirement? So you need to identify, and also you can use design of experiments to optimize the process to select. So if we consider sustainability, so typically in most of the process, they use sugar or glycerol or some high value nutrient. High value means it's mostly, they're all sometimes ready-made media. It's expensive. So the opportunities are, probably in India, we quite a lot of time we see fungal-based contamination, not only in laboratory, even if we go to any market, if they dump waste, we can see quite a lot of activities, and so fungus, it's quite it's a predominant in most of the places where waste fruits or vegetables stored, or even if they leave it now after 2 or 3 days, you can see these issues. So clearly so it's not a problem for fungus to grow in certain nutrient. So clearly for example, outdated fruits and vegetable from the supermarket, it's a huge issue. So when you visit now, probably first a couple of days, it'll be fine. After 3rd day or 4th day, here in supermarket, they can keep the fruits for a week or 10 days because the environment, it's cold weather. Sometimes apples, if you leave it on the table, it stays for 3 weeks. It looks same after 3 weeks. Whereas in India, it's after 2 days because of warm weather and the climatic condition, it starts to decay very quickly. So after 2 or 3 days, all these perishable fruits and veg, mostly a lot of waste issues. So those kind of waste, if it can be identified well in advance, if it's processed to extract the sugars from the material, then that will be a fantastic medium for these kind of processes. So you don't need to look for glucose or some, the value. There is a cost associated with it that could be reduced, and also it will give you give another, give a solution for another issue, waste issues. So you can consider, and also agricultural waste, for example, sugarcane is predominant in India. It's any residual sugar residues from sugarcane. They may have sugar, residual sugar that can be used in these kind of processes. As we know, fungus grows well in our climatic conditions. Clearly, the fermentation process, if we set up in aseptic conditions, you'll be able to achieve this. And this case study, what you have seen, it's from Fusarium venenatum, which was isolated from soil, one of the agricultural soil in local environment. So there are plenty of opportunities. But before we, we chip into market as a product, it needs quite a lot of research. And these companies, it started nearly I think 20 years ago. So they have been doing so many process optimization and to make sure that particular fungus or all the RNA content from the fungus removed in the process. So it takes time, but it's worth. That's finally they're selling these kind of product around the world. I think their ambition is to reach out around the world, and so clearly countries like India, it's a fantastic vegan based protein, and currently soy meal is available. Soy meal I have seen in many food in India. It's used, so you may have number of other options if these kind of fungal based protein come to the market, and it has a unique taste. I have tasted it. It's fantastic. It's really nice, so no problem. When you compare to the soya meal, or in India, I think it's sold as a meal maker or something. So I have tried and I have compared. For example, if you make a meal maker vegetable biryani or something, if you use this fungal based protein, almost same. And the taste also sometimes, if those who are in vegetarian, so probably they never tasted chicken or anything. So for them, it's a fantastic alternate. But those who are already practicing as a non-vegetarian or meat eaters, so if for certain health issues, they should not eat meat, or because for example, it should not mean they have to reduce their intake. For example, hypertension patients with hypertension or cardiac issues, they have issues of taking meat. So for them, as I have shared in the beginning, the fat content in these final products, it's extremely low, but the taste, it's almost it's a good alternate for meat. So for them, it's fantastic. So they have another option, because it mouth feels like a fibrous protein, and you can make number of shapes and size for the customer's need. So that's a long term. There are so many options available related to the customer focused products. But for the process side, if you're a biologist or microbiologist planning to do research on this, as I mentioned, it's process optimization it's a key. We need to consider, we need to think very carefully how to avoid contamination and so on. And also in fermentation process, it needs certain things. For example, how to maintain dissolved oxygen tension, the pH additions and the nutrients. That's an optimum, how to provide the optimum conditions for the best process outcome. Just to capture now this harvesting technique, this is self-harvesting. Probably in our labs or in biology labs, microbiology lab, you may have a small centrifuge. So, whereas if for industrial scale processes, though, you may not be able to use those kind of 15 ml, 50 ml centrifuge tubes. We need industrial scale process. So that means as I mentioned, it's a continuous fermentation process. So that means the process happening continuously. So you need to extract the culture continuously. So here they extract and then it passes through this decanters. So in decanters, when the culture is supplied, so it's continuously rotating. It's almost like a scraping type of approach. And the biomass, if it's all deposited then on the walls of the cylinder, but the cylinder now continuously when it rotates, it's like a spiral, screw type of approach, then continuously it removes the biomass so that we can separate the cell paste, and remaining residual or cell-free supernatant can be separated. There are a number of designs available for industrial scale separations. And as I mentioned, it's a degassing. So degassing is important in this process because it's a airlift fermentation process. Or in this, we continuously gas is supplied. For example, air and gas. So we need to release the gas to avoid any of the pressure build up in the bioreactor. And the heat shock. So another process used in this mainly mycoprotein production. So heat shock stage, cells are repeatedly heated at 60 degrees Celsius. So this causes the lyse and releasing RNA. Because RNA content in fungal hyphae, that would be, if the RNA is not removed effectively, then there are certain issues. The final product may not be good, or it's not suitable for consumers. So the high RNA intake, which will cause some health issues. So we need to minimize the RNA content to less than 2%. So I think it's a heat shock process will help to do that. And then this slide already I have shown now. Then finally it is processed and then finally make it in various forms for the final product. It's in the local market, we can get crispy nuggets, roasted slices of fillets, or so these are all based on local needs, local food habits. For example, in the UK, these are all quite common approach. For example, mince or sausage in morning breakfast, or if it's a if you know British breakfast means it's like a few pieces of tomato and also a few greens and a couple of pieces. It's a combination of vegetable, potato, and sausage, and some greens, tomatoes. For that, it's excellent. These sausages, nuts, it's a good replacement for a meat-based one. And mini savory eggs and mini. So there are a number of products coming to the market. So you can think of, it's probably someone, those who are listening, we can put some in chat box. What sort of product in India can be replaced, for example, using these mycoprotein? So when my thought now, initially I thought maybe in vegetable biryani, if they are making with soya chunks or meat or chicken, we can use these chunks. For example, it's a good to remove, instead of meat, we can use this to get that done. If you think any other alternate, please include the detail. Your suggestion in the chat box. I'll look at it. So what are the options are suitable? Because India is it's a even though I studied in South India, it's a small, it's only one part of it. It's a huge. Each state we have our own food habit, and the taste different. If you move from south, for example, in South India, some food are hot, or add some chili and combination, lemon, coriander combinations in some food. That's when we move towards the north. It's when you cross from Chennai to Hyderabad. So I have traveled many times up now for my the PhD research fellowships and conferences. It's a variety of things happening. It's colors and so many things. Even food variety. Each station, when we cross from Chennai to Hyderabad, and then when it goes through UP, MP, and then Delhi, before you reach, you'll see like hundreds of food varieties sold in train stations and all. But if you go to the local market, there are so many, the indigenous food practiced or used in each state, or sometimes each towns and cities. They have their own special varieties of food items. But think about in a bigger picture of these kind, for example, if it's a meat substitute, so meat-based product, also number of products available in each state and cities and towns. But if you create one unique product and scale up to the industrial scale, then look at the opportunities. So around the country, you can supply and create a big brand. So it's a huge opportunity. Because even though if they have their own, the set of food or anything, but some of the base, if it is a meat replacement, so this is a good option they can choose. So, yeah, and also it's a good for parties to make various food items. It goes very well with noodles or rice, and a combination of spices we use in India. It clearly it works well, so it's not a problem. Because another thing I have to highlight, if some of you, if have been to UK or Western countries, sometimes in the US also, most of the food items, here we eat, it's almost like you may not feel that much taste, but only the coating that will have those flavor, but inside it may not reach, or it's. Whereas in Indian food, most of the food it's cooked with various spices and flavors and ingredients that infused with the whatever the ingredients you put. For example, if it's a meat or even if it's a soya chunk, so in India we expect all the ingredients should reach, or it should be uniformly mixed in those chunks. Then only we feel that, yeah, it is nicely cooked or it's ready to eat. Whereas in most of the here, not mostly like European food at all, if you see, it's just coated on the surface. If you eat it inside, that will be still you feel the raw, or it's not raw, it's cooked, but it doesn't, those ingredients, it never reach inside. But it's so in this approach, clearly the mycoprotein that's what I would like to say, because of they have the fibrous nature, because it takes all the ingredients and flavors into the chunks, and clearly it will provide it and give the best taste output, or you feel basically at the end you feel satisfied. Probably it's India, it's 12:30. I think most of you might have finished your lunch now, so it's a Sunday, so probably maybe next Sunday you can try. I'm not sure whether it is available in the Indian market. Maybe you can start with the soya meal chunks, how it works, maybe, and then you plan to start something in research. I provided a couple of research papers related to the fungal protein. Those who are doing projects or masters or PhD projects, maybe if you're interested, you can look at it. And there are a number of research articles available for fungal protein. And also I'd like to highlight in Europe, so this company, it's Quon, it's big brand here. You can see almost all the supermarket, this product. It produced in thousands of tons per week. It's very large fermenters they use to produce this. And now various companies, startup companies in Europe, they all working on various fungus, because this is only one, Fusarium venenatum. So a number of fungus, a lot of work going on, and they are getting some new taste, new flavor, and a new product to the market. So, and also in a bigger picture, they are also getting very good investment for their businesses to grow. As I mentioned, these trends, it's a replacement of a meat, means meat industry is the largest one, I would say, globally. A lot of countries, they're all using, and if you produce a product which could replace meat, then you have global access. It's huge. But yeah, so I'd like to share that. And if anyone interested in future related to this for a startup or any businesses, I'd be happy to support and then to see how I can help or I can contribute or we could help in the process development or startup process. So with that, I'd like to stop it. So this is the title we started. And thank you for listening. And I have asked to provide some local food items in India, which could be, we can think of for introducing these mycoprotein approach. I'll see if any information available in the chat box. So I'll stop the slide share and then so ready. Okay, I'll go from the top. Yeah, shawarma. That's a very good suggestion. Shawarma, here I think it's a Middle Eastern food. So pita bread, and then inside a combination of vegetables and meat. So yeah, we can target. That's perfect. Shawarma or that's good. Then samosa and momos. Sorry, I don't know what is momos. This is the beauty of India. I know samosas. Even though I grew up, I studied, I have been visiting India for almost every year. So I'm sorry, I don't know about momos. Maybe if someone might send another text to explain what is momos, that would be great. And then excellent. Thank you. Thanks for your appreciation. Individual studies in Indian culture and go to abroad. Probably, that I may need to organize another event like this to share those kind of information. That's from Mr. Mohammed. And then Mohammed Kudus. Thank you, sir. Excellent work. Protein shake. That sounds good. Protein shake. As I mentioned, it's a protein. It's an excellent nutrient combination. Mycoprotein. If you remove RNA content, it's fantastic. To create a protein shake, it is possible. It's because a lot of people, around the world, everyone wants to be healthy. Mainly healthy, and they start to go to gym to maintain the health condition, and also they used to take a lot of protein supplement. So protein shake would be good. Sometimes that will go, protein shake in liquid if it's I need to think. There are a number of options in protein shake. For example, if it's a tinned one, or are you going to provide these kind of chunks for the, so those who are making milkshake in the community? You might have seen lassi and so many things available. But if you provide very clean protein to them, it'll be a fantastic market. Yes, why not? They can use it, or we can create a protein shake. That's excellent. Spreads. Spreads is good. So here we use spread in between sandwich. So sandwich is egg sandwich and so many chicken sandwich. So this replacement, if you make it a paste, that paste could be spreaded on the bread. So instead of chocolate spread or chicken spread, why not? We can use this spread. That's from Dr. Nivedita. Thank you. Tofu from Bushra Ahmed. That's good. Tofu you can create. Exactly. Tofu, they're making that with soya. So it's we can replace but I'm not sure tofu market in India, but tofu has a great market in China, Japan, most of the Southeast Asia. So we need to think in that aspect. Now, it's not just even if you make some product, if it's not working or to the local, if it's not accepted or suitable for the local market, but some of the products it has a global reach. So under the because of various global agreements and all, transportation, everything is it's effective. And there are various large chillers are available to transport these material from one country to other. For example, in the UK, we are getting tons and tons of food products from Europe. So almost every day, there are around thousands of trucks coming from Europe to here to distribute various chilled food, fruits, flowers, whatnot. Everything is coming. And also similarly, quite a lot of meat product meat and milk-based product going from UK to other parts of the world. So it's a transportation, it's not an issue. Momos are kind of a dumpling. Oh, fantastic. Thank you. Yeah. So momos would be, maybe dumpling. Yeah. Then you may need another base. It could be, yeah. Maybe I need to check how it looks like. I can't predict. Maybe I'll do some research about momos later. Veggies. Okay, that's good. So Chinese food, yeah. Because actually in India, so we have variety of flavors and so many things available, but it's easy to reach out because most of the states we have that tendency. If something new, why not? We will try. So when you have that approach, it's easy to enter into the market. That's how probably if you think about pizza, burgers, according to our own food varieties, we cannot compare. It's not at all reachable. It's all so not that much taste, and it's a high fat content. But it's almost in every cities we can see McDonald's, Burger King, and all sort of Western branded stuff in Indian cities. So they reached because people want to try. Sometimes they ignore about the health. But if you put a tag with health tag and a protein supplement and a good replacement for meat, so here you can reach out both vegan or vegetarian community, and also those who are eating meat, but they want to go towards something healthy option. This is a fantastic one. So in that case, now all these Chinese varieties could be introduced. Thank you. What else I have? Can't be the best supplement for malnutrition conditions. Yes, you can. It's a protein supplement. Beans are good source of protein. Yeah, we have that's in India, that is from Gibson. Yeah, thank you. Yeah, because in India, that's probably most of us know, even wherever we go. Definitely in the past 20 years now, I've been traveling. I've seen early days, maybe 1990s. When you go, you can see people are struggling. I don't know, for some reason, some little bit of poverty. But nowadays, everyone can afford things, and mostly they're happy. Occasionally, we can see few, the poverty, but it's all minimal. So protein source, in most of the cases, it's pulse, beans, number of bean varieties. It's great. And it's already we have been using in our food. For example, lunch. In South Indian culture, we use various curries. Mostly, I think they'll have a chickpea or something, protein already in that. So it's not a replacement. If already there are some food in that system, it will be in addition to that. You can provide these meat supplement. That's what I would say. Yeah, thank you. Yeah, that's very good point. Yeah, and yeah, that's you can also think there are so many options. If there are protein which can be taken, even the dal we have, so dal also contains protein. So we need to think how, if these protein are a nutrition amino acid content, we need to look the features of these mycoprotein, and then you need to highlight or pitch to the customers. Sometimes, if already some product available, enter into the market will be challenged because people have some answers to that. So how you are going to convince to replace their food habit, unless if there is a health-based ground, or it's something which we need to convince, or it should be cheap, it should be available everywhere. So that kind of approach, I'll leave it with the audience, those who are listening. Right, can't be used in animal feed like pedigree for pets. That's fantastic. Yeah, that is possible, because I think that is a global reach out for some in the pedigree of dogs now. Because in India we have, in Western country we can't see any. Western means in UK. So far I have been here, for the 22 years, I never see any stray dogs. Yeah, so mostly it's all fantastic. They all, well, it's almost like a family member. So they provide the best food for their pets, and they don't mind the cost, whatever the cost are put label. It's always they want to provide the best. So similarly in India also, in India it's a diversity, it's a huge. Some challenging, some of them they can afford, some of them they can afford anything. So you can target now. Sometimes we need to consider these kind of product, one for premium product, high-end product. Some of them they can afford, and the reach out. Now the population is about 1 billion people, so you can, even if it is a small fraction, 100,000 people they use, still you'll be able to sell 100,000 pockets or packs. That is sufficient, something like that. So you need to think much more bigger way. Yeah, for pet food, it's a great. Yeah, it is possible. Soil quality affects the quality of mycoprotein. Is soil quality affect the? Oh, it's that's another tricky question. What are the lab tests required to test this mycoprotein? Probably those lab tests means first make sure I'm following the right questions here. Yeah. So it's a protein requirement, and maybe because of the time limit, now if I expand that answer, I can't answer everything. I would recommend, please go through the research article link I gave, and also do some research on mycoprotein. There are so many reports available. What are the tests you need to conduct? For example, if it's a nutritive value based, what are the amino acid profile you need to look for? What are the essential, non-essential amino acid, and so on. So to get, if it's you need to think in those lines. Maybe you need to do some research rather than explaining it. I need another lecture for that. So in soil quality, is soil quality affect the quality of mycoprotein? I don't think so. How? Because if we are isolating a fungus from soil, that's and then mostly it's all processed in a fermentation-based process, and then finally you are getting a product which is a protein-based, and it goes to the consumers, and any of the residual wastewater, if it's treated properly according to the local regulations. So you need to look at the environmental agency, mainly in India, Indian environmental agency regulations about wastewater treatment and all. If you design I don't think it will affect any soil quality, unless if yeah, if any specific question, maybe in general, it's fine. Can we commercialize mycoprotein tonic for diabetic patients? That's excellent. Yeah, because it's a protein supplement. If you know any tonics already available, maybe I would recommend please have a look at it and then how you can replace. It may not be tonic, it could be like a protein supplement, nutraceutical. Well, yeah, with nutraceutical type of approach. But that all of them know when, for example, food, it has a specific regulatory and so on. For cosmetics or for nutraceutical or any supplement or sometimes any biopharma, they all, you need to look at the regulations associated with that before you think what sort of application you can target. Yeah, then, okay, I think this is helpful in maintaining cholesterol levels in blood. I think one of the slide I have shown, please have a look at it. Yes, it is, because fat content is low and it's mainly the protein with spectrum of good amino acids. So it is, maybe you may need to do further research. For example, if we are finding, looking for a new innovation or anything, then you need to find another source of, or source means which fungus, which could be used, that could be used to develop the process and so on. You need to look in detail how to approach that. Yeah, cholesterol level and all. Yeah, you need to look at those details. That specific questions. With that, I think I'm all right. Now it's about 15 minutes questions. So if anyone got any startup interest, if you have a commercial interest, also I'd be happy to do like separate discussions. We could get in touch with the organizers and we'll see. Startup means then it's you need to have some basic knowledge and funds needed. So there are some challenges associated with that, but it's possible. But I cannot, provide everything. So maybe if you already have a lab, microbiology lab with the fermenter, then if any fungal related work happening in your labs, or the fungal, so you can grow and then you can ferment and so on, then probably you can start something basic. And then if someone in the lab, if already they have good fungal culture collection, then maybe you can look at it, how to do some screening, initial screening, to find out what type of fungus are available. Is it non-pathogenic? Is it good fungus? Because at the end, for any of the commercial product, where it comes from, that organisms. Are you isolating that organisms from sewage? Then probably it's very hard to sell the product. If you are going to isolate the product, that fungus from Himalayas, or best part of any of the world, then so you can brand very nicely, even Himalayas Mycoprotein, something like that. People will be happy to see, even though it's just a fungus isolated, the vision, they will think it's a pure, pristine, all the ice crystals, straight away they'll come. So you need to think in those directions. So where it comes from, it's very, very important. Our soil would be fine, agricultural soil, fine. So yeah, please look at those things. And also you can think about pathogenicity and what are the good fungus. Is it possible to isolate some best fungus? Because in the UK or in Europe, we get a cheese called blue cheese or green cheese, various colored cheese. One of them, definitely, it's a fungal-based cheese. It's already they're using live cultures and live fungus. We can see the fungal hyphae growing on the cheese. And then during Christmas days, everyone enjoy eating those kind of fungus, goes in the system, no problem. Is it possible to get those kind of good, it's already tested and now millions of people they have been using for parties and so on? Why not? Is it possible to get something like that? It's easy to sell. So with that, I think I can stop it now. Almost I can see around 100 participants in this room, and I'd be happy to meet you all some other time in some other occasion. But thank you for listening. Thank you, Professor Deshmukh, and also
Yes.
the
Excellent
organizers. Session. Yeah, that's thank you very much. Thanks. Yeah, thanks for listening.
Thank you, sir. Thank you. Yeah, Nagargam ma'am, we can go for conclusion and vote of thanks.
Yes, sir. Yes, sir. So I request Kiran ma'am to please proceed for vote of thanks.
There are a few questions in the chat box. You can read that questions.
Sir, already read, sir.
Okay,
the question answer session is over, sir.
Okay, no problem.
Yes, sir.
Good afternoon, everyone. I, Kiran Jagtap, Assistant Professor at Sanjani College, feels a great honor to propose the vote of thanks on behalf of Department of Microbiology, Sanjivani Arts, Commerce and Science College, Kupura. First of all, I would like to propose hearty vote of thanks to our distinguished speaker, Dr. Patnath Rahman, for giving his valuable time and guidance and making this webinar interesting and meaningful. Thank you, sir. Next, I would like to thank respected Dr. A.M. Deshmukh, sir, President of Microbiologist Society, India, for providing us such a great platform for this interactive session. Now, I would like to express our profound gratitude to our principal, Dr. S.B. Dhaikar, sir, for his support and guidance. I would also like to thank our beloved HOD, Dr. S.A. Bhutara ma'am, for her guidance. I'm happy to express vote of thanks to our staff members who have made this webinar a success. I also thank all the respected attendees joined through online mode for their presence. Finally, the wonderful students who have been a patient listener for entire session. Thank you so much for your cooperation. Once again, I thank all for your cooperation. Thank you, everyone.
Once again, thanks, Doctor. We'll meet you soon in next session.
Thanks, Dr. Daika. Yeah, that's fantastic. So your communication was excellent. To organize this. Thank you very much. I look forward.
Thank you. Thanks. Yes, sir. Thank you. Thank you. Have a good day. Bye-bye.
Thank you. Bye now. I'll see you later.
Bye.